Hi, everyone, and thanks so much for joining us today. Whether this is your first time attending or you've been with us since the beginning of this series, welcome. We're excited to have you here for session 3, where we'll be diving into the principles of mixed mode chromatography. And before we start, just a couple of quick notes. This session is being recorded, and you'll be able to find the full recordings as well as recordings of session 1 and 2 on our YouTube channel. And if you have any questions along the way, feel free to drop them in the chat. We'll have time also for the live Q&A at the end of the session. And before we get started, a quick word about who we are. We are a chromatography company based in Chicago area, focused on developing innovative HPLC columns and compact mini HPLC systems. And with over 2 decades of experience, we also provide custom method development and technical support to help solve complex separation challenges. And if you ever need a fresh approach to an HPLC problem, or you just want to explore new column options, don't hesitate to get in touch. We are always happy to collaborate. With that, I'll go ahead and share my screen so we can jump into today's topic, Introduction to Mixed Mode Chromatography. Good afternoon, everyone, and welcome to our third session in HPLC educational webinar series titled Introduction to Mixed Mode Chromatography. I'm Jovana Mitrovic, an R&D chemist at Silk Technologies, and I'm excited to share with you a technique that has changed the way we approach complex chromatographic separations. And whether you are working with pharmaceuticals, biological samples, or food products, mixed mode chromatography can offer elegant solutions to difficult problems. In our previous session, Mastering HPLC, we focused on the hardware components of an HPLC system and their influence on separation quality. We also discussed how to select appropriate detectors based on analyte characteristics and application needs. A big part of that session was dedicated to choosing suitable columns and mobile phases, understanding when to use gradient or isocratic elutions, as well as how different factors affect efficiency and resolution. We closed with an introduction to method development with a brief step-by-step guide. Today, we'll take a step further by introducing mixed mode columns as tools for solving more complex challenges. Here is what we'll explore today. We'll begin by examining the limitations of traditional single mode chromatography methods, like reverse phase, ion exchange, and normal phase, and why they often fall short. Then we'll introduce the concept of mixed mode chromatography, what it is, why it works, and how combining interactions improves performance. We'll break down the types of interactions at play, discuss key benefits, and look at how mixed mode columns are designed. Then we'll move into applications, focusing on our PrimeSep and Nucrom line of columns with case studies and example separations. And finally, we'll preview what's coming in our next session, which will focus on practical method development using mixed mode columns. So let's start with what we are all familiar with, traditional chromatographic modes. In normal phase chromatography, retention is based on polar interactions between analytes and a polar stationary phase. Reverse phase is the opposite, relying on hydrophobic interactions with a non-polar stationary phase. Then there is ion exchange, which separates compounds based on their charge. Each of these is effective for a narrow range of compounds, but real-world examples often contain analytes with multiple properties, some polar, some non-polar, some ionizable. And that's where problems arise. So each of these traditional modes are limited to by single type of interaction it uses. Reverse phase can struggle to retain polar or charged compounds without any hydrophobic property. Ion exchange fails when dealing with neutral species. Normal phase is incompatible with aqueous samples and sensitive to very small changes in water contact. These limitations lead to poor selectivity, difficult method development, and lots of time spent switching between columns, reoptimizing mobile phases, and hoping for resolution. And as complexity increases, single mode approaches become increasingly inefficient. So mixed mode columns solve these problems by integrating two or more retention mechanisms into a single stationary phase. You might have reverse phase retention working alongside ion exchange. You can incorporate multiple interactions from hydrophobic, pi-pi, hydrogen bonding, or ion exchange in a single column. And this gives you more control over how different analytes interact with the column, making it possible to separate very different molecules or very similar ones in a single run. And you can gain also selectivity, sharper peaks, and flexibility in mobile phase composition, and also including compatibility with LC-MS system. It's all about enhancing efficiency while simplifying method development. And what's important, mixed mode columns can also streamline method development. Instead of screening multiple columns and mobile phases across reverse phase, ion exchange, or HILIC, you start with one column and gain selectivity from how you adjust mobile phase parameters. It's a more efficient path to robust methods. Single mode methods force you to treat complex analysis as they fit neatly into one category, polar, charged, or hydrophobic. But many compounds don't behave that way. And relying on a single interaction often means overlooking key retention behavior. And traditionally, secondary interactions were seen as problem. Think about peak tailing from silanol activity. We were trained to avoid these effects. But with mixed mode chromatography, we intentionally design stationary phases to harness secondary interactions. And instead of eliminating them, we control them. This gives you three key advantages. We can fine-tune selectivity, achieve better reproducibility, and extend the stability of the column over time. So question is, how do we build a mixed mode column? There are several strategies, and here are just a few visual examples. One approach is to embed ionic groups into a hydrophobic ligand positioned near the silica surface. Alternatively, the ionic groups can be placed at the terminal end of hydrophobic chain. Also, aromatic rings can also be introduced to enable pi-pi interactions. In some designs, we can have both acidic and basic functional groups incorporated into hydrophobic chains. And by adjusting the density, strength, and position of these functional groups, we can fine-tune the column selectivity and retention characteristics. The result is a highly versatile chromatographic system, and essentially a customizable toolkit that enables you to achieve separations that single-mode columns simply cannot. Additionally, the geometry of both the stationary phase and the analyte plays a critical role. The placement of charge groups, whether near the silica surface or at the end of the long ligand chain, can influence how easily analytes access and interact with them. And similarly, the analyte's own shape and the location of its charged regions can play a big role in selectivity. This, let's say, spatial compatibility affects interaction strength and selectivity, and especially important for large, bulky, and rigid molecules. And we'll see some examples in the next slide. So let's start with prime sep columns. This slide gives you a clear visual summary of different prime sep columns and how they're grouped based on their ionic functionality. On the left side, we see the acidic columns such as prime sep A, 100, 200, and C. These all contain acidic functional groups embedded within a hydrophobic ligand, typically a C12 chain. As we move upward on this list, the strength of the ionic interaction due to pKa increases, offering a greater retention for basic analytes through cation exchange mechanism. On the right side, we have the basic columns, prime sep SB, B, D, and B2. These columns incorporate basic functional groups, making them suitable for retaining acidic analytes via anion exchange. Again, the strength of ionic interaction increases as we move upward. So what's key here is that all of these columns maintain a reverse phase backbone. So you can separate analytes based on both hydrophobic and ionic properties in a controlled manner. And this structure allows you to fine-tune selectivity depending on your analyte's charge and hydrophobicity. Now, let's look at this. Prime sep columns, as we mentioned, are reverse phase columns with embedded ionic groups. There are three main parameters you can control to adjust retention. First is hydrophobic interaction, which is tuned by the organic solvent content in your mobile phase. Second is ionic strength. By adjusting the buffer concentration, you can modulate ion exchange interactions. Third is pH, which impacts the degree of ionization of both the analyte and the stationary phase. And these three axes of control, hydrophobicity, ion exchange, and ionization create a versatile, tunable platform. So now let's look at specific examples of prime sep columns in action. Prime sep 100 and 200 are both C12 reverse phase columns with embedded acidic groups, making them excellent for retaining basic compounds through cation exchange. The key difference is the strength of these acidic groups. Prime sep 100 contains a very strong acidic group, pKa around 1, ideal for analyzing moderately basic compounds. Prime sep 200 has a slightly weaker acid, pKa around 2, which works well for analyzing more strongly basic analytes. These columns also retain neutral hydrophobic compounds through reverse phase interaction and separates also acids via ion exclusion mechanism. Here is an application example. Anthocyanins are natural pigments found in fruits like grapes and berries. They are charged, structurally very similar, and sometimes challenging to resolve. Using PrimeSep 100, which combines hydrophobic and strong cation exchange mechanism, we achieved excellent separation. The C12-like chain provides hydrophobic retention, while the embedded acidic group enables fine control over ionic interaction. And this application is relevant to food analysis, supplemental quality testing, and plant-based research. So now let's just take a look at an application involving several plant growth regulators, including cytokinins and related compounds such as thiadizuron and brasinazole. The four cytokinins such as zeatin, kinetin, benzyladenine, and isopentyladenine share a purine ring structure with slight modification in side chain. This structural similarity makes them challenging to separate using traditional methods. On the other hand, thiadizuron and brasinazole, while functionally similar, is structurally very distinct from these cytokinins. And all these compounds differ widely in their ionization behavior, polarity, and hydrophobicity. PrimeSep 200 enables the effective separation of this chemically diverse group using its mixed mode design, featuring, again, hydrophobic C12 chain and embedded acidic group. This acidic group can interact with slightly basic regions of these molecules, enhancing retention and selectivity, while the hydrophobic chain helps retaining nonpolar features present, for example, in brasinazole. And by adjusting the mobile phase, we can selectively enhance interactions for different compounds based on their structure. And this versatility is ideal for researchers analyzing multiple hormone classes, whether in a plant biology, agriculture, or environmental analysis, without requiring multiple, for example, chromatographic methods. We can retain diverse chemical compounds, and, for example, less hydrophobic will elute earlier, and by increasing the acetonitrile and doing the gradient, we can elute more hydrophobic compounds. And one of common issues in, for example, agrochemical or botanical analysis is matrix complexity. Soil and plant extracts contain many interfering compounds, such as can be amino acids, sugars, pigments. And mixed mode chromatography can selectively retain and separate the active ingredients even in the presence of this interference. For example, with PrimeSep 200, you can isolate and quantify your targets while pushing matrix components out either earlier or later by adjusting the mobile phase composition. Now, moving on to basic stationary phases, PrimeSep B and B2. These columns, again, have embedded basic groups, offering anion exchange capability. PrimeSep B features a strong basic group with a pK around 10, which is ideal for separating, let's say, weakly acidic compounds. PrimeSep B2 has a weaker base with pK around 7, and that's better suited for a strong acid. Both still retain non-polar compounds using reverse phase interactions, and they're fully LCMS compatible. Now, let's consider the analysis of vimseltinib, a basic hydrophobic tyrosine kinase inhibitor, which was recently approved by FDA. On traditional columns, compounds such as these often exhibit poor peak shape due to ionic interaction with residual silanol groups. So this embedded basic group in PrimeSep B provides cation exclusion effect and helps shield silanols, reducing unwanted secondary interaction. The result is sharp, reproducible peaks, making it well suited for LCMS and drug development workflows. There are two mechanisms at play here: hydrophobic interaction and cation exclusion. And by adjusting the buffer concentration, like in this case ammonium formate, and organic solvent contact such as acetonitrile, we can influence the retention of this compound. And for example, reducing the amount of buffer, in this case, increases the repulsion between the stationary phase and the analyte, which decreases then retention. Pigtailing is a common issue when analyzing basic compounds or traditional silica-based columns. And this occurs because residual silanol groups, those weakly acidic sites on the silica surface, can electrostatically interact with basic analytes, leading to distorted peak shapes and retention variability. Prime-sep B and B2 columns address this problem by embedding a positively charged group near the silica surface. And these designs offer like this dual advantage. It enables controlled anion exchange retention for acidic molecules, while simultaneously shielding silanol groups, resulting in improved peak shape for basic hydrophobic analytes. And then, consequentially, ionic interactions that cause pigtailing are minimized. So, for example, when analyzing, like we have here, oxymetazoline on the mixed mode prime-sep B2 column compared to traditional C18 reverse phase column, the peak shape is significantly improved with prime-sep B2. The result is consistent, sharp peaks, and reliable retention, even for challenging basic drugs and amine-containing analytes. This makes just prime-sep B series columns ideal for the analysis of hydrophobic basic compounds. And before we introduce now the Nucrom column series, let's just take a moment to understand what makes their design fundamentally different from other mixed mode columns. In the prime-sep line, the ionic functional groups are embedded close to the surface of silica. Nucrom columns, on the other hand, position these charge groups at the ends of long, flexible ligand chains, far from the silica surface. And in a moment, we'll see why this design choice is significant. The Nucrom series are also divided into two main types, acidic and basic columns, and each type comes in two versions, one with a longer alkyl chain, such as Nucrom A or Nucrom B, and one with shorter alkyl chain, such as Nucrom AH or Nucrom BH. And thanks to their flexible ligand architecture and innovative charge positioning, Nucrom columns are particularly effective at separating highly multi-charged compounds. In traditional ion exchange columns, multi-charged analytes often interact non-uniformly with the stationary phase, leading to poor peak shape and inconsistent retention. These issues arise from the fixed surface-bound nature of this ionic group, which can limit accessibility and hinder uniform interactions. Nucrom columns address this challenge by positioning ionic interaction sites away from the silica surface. And by attaching charged groups to like these flexible ligands, the column creates a more, let's say, solution-like environment, allowing for ions to interact more uniformly, similar to their behavior in free solution. And this results in improved peak shape, greater retention consistency, and more, in general, more reliable overall analysis. this spatial repositioning of charge is critical. And by distancing the ionic groups from this rigid silica surface, neutron columns reduce this steric hindrance and enhance accessibility, especially for analytes with complex geometries or buried functional groups. And the result is constantly better separation performance for these challenging, highly charged analytes. So here is an example with ADTA, a highly charged chelating agent with very poor UV absorbance and strong affinity for metals. ADTA doesn't retain on a typical reverse phase columns. And it's also hard to elute on a single mode ion exchange columns, and often can also bind metal components in HPLC system. Here, we used iron complexing in order to make it suitable for UV detection. So we detected at UV 260, and with neutron BH, we also achieved excellent retention as well as solution using an aqueous mobile phase with just 2% organic and a small amount of sulfuric acid. And this long flexible ligand enabled just like a good interaction with ADTA with an excellent and narrow peak shape. Another excellent application of neutron columns is the separation of nucleotides such as AMP, ADP, and ATP, which they differ only by single phosphate group. And on conventional single-mode ion exchange columns, these compounds often produce poor peak shape and limited resolution. Neutron B utilizes a combination, again, of this reverse phase and ion exchange retention, and its charged functional groups, which are located far from the silica surface, allows for consistent and uniform interaction with these highly charged analytes. And even using a simple isocratic method with just 10% acetonitrile and 0.1% sulfuric acid in the mobile phase, neutron B delivers sharp, symmetrical peak and baseline resolution. And this just makes it an ideal choice for biological or pharmaceutical workflows. To recap, mixed-mode chromatography provides a powerful solution for complex separations by combining multiple interactions in one column. We reviewed how PrimeSep and neutron columns overcome the limitations of single-mode techniques and explored some real-world examples that can be used in pharmaceutical, botanical, and biochemical applications. And in our next session, we'll focus on practical method development with mixed-mode columns, including how to balance retention, resolution, and sensitivity, plus some very difficult case studies that show real success in challenging applications. Thank you again for joining today's sessions. If you have questions about the content or need support selecting a column or want to discuss your specific application, just please free to reach out. You can contact us at Silk Technologies through our website, via email or by phone, and we are always happy to help you achieve your separation goals. And we hope to see you in the next webinar. Now we're going to go to Q&A session. Just please feel free to ask any questions you might have related to this or your own analysis.